next talk, I will give you guys some reminders. I'm Ana Mota, and I will be the moderator for this session, which is Concurrent 1. And this session is implementing the use of digital data in basic biodiversity research. I already say if I pronounce your name and if wrong, and if I use the wrong pronouns, feel free to correct me at the beginning of your talk. Just some reminders. You already were here yesterday, so you know this, but doesn't hurt. For the virtual audience, the questions should be entered in the Q&A dialogue. And for whoever is here, just raise your hand at the end of the talk, and we will bring you the mic. And people, the virtual audience can also raise their hand on Zoom and ask the questions if they don't want to write it on Q&A. The speakers have 10 minutes for presentation and 5 minutes for Q&A. And the session will conclude at 15-minute mark. So at 8 minutes, I'll be sitting there. At 8 minutes, I will stand. You see me. I'm wearing green, so it's easy to see me. And at 15 minutes, I will have to stop you, whatever you are. So, and I think that's it for the reminders. Our first, supposed to be second speaker, will be Joseph Mohan, and the talk is Consolidating Diatom Ecology and Trade Data to Decipher the Hieroglyphic Forms and Enigmatic Functions. I'll ask you to wait just a minute so we can start exactly at 10:45, but you can come and maybe test the pointer and stuff if you need.
Good. Okay, great. Thank you, everyone. I'm Joe Mohan, a postdoctoral scholar at the University of California, Irvine. I'm one of the Biodiversity Data Capacity Fellows, so thank you to the conference and Symbiota for bringing me out here. And thank you to my collaborators and my lab group. When I gave this talk on Wednesday to them, it was about 13, 14 minutes, but I think I've had enough coffee. We should cut that down a couple minutes. So I'm here to tell you a little bit about diatoms, sort of sometimes the forgotten collections in many herbaria. Just talking with a couple recently, it's like, oh yeah, I think our herbaria has, somebody put some diatom slides somewhere and maybe we've forgotten where they are. So I'm really hoping to consolidate a lot of that information and giving this talk as sort of a pitch for an idea to put together a lot of these components. Oh, and also thank you to Jen Slouch, a PhD student in our department, for putting together these nice, wonderful diatom hieroglyphics. So if you don't know diatoms, they are maybe the coolest organisms out there, in my personal bias. They're single-celled algae, they're photosynthetic, and what's really interesting about their morphologies is that their traits are tied to this outer cell wall that is made from secreted silica glass. So because of that, they make really nice environmental indicators because each species has pretty narrow niche, and then each species has a unique shape and size and form and a bunch of traits that go along with that that niche. And because they're made of silica glass, they fossilize really easily and make wonderful paleontology tools. If you're not convinced yet, they're a major primary producer. So we like to say like every third or fifth breath, just thank a diatom for all that wonderful oxygen. If you're still not convinced, another one is that a lot of the small breweries around the country, when they started, used diatomaceous earth filters to filter their beer. So if you had the lager last night, maybe not anymore, they probably use an RO system, but when Free State opened in 1989, it was probably a diatom-based filter system. So thanks, diatoms. So what we have in diatom land and a strong community of diatom scientists is we have really great consolidated taxonomic nomenclature, mostly from algae base, if you're familiar with it. Query and Query have about 170,000 species. About almost 17,000 of those are diatoms, and this is really nice because it links basionyms, synonyms, homonyms, everything. We can pull that data and go like, oh, Sherry Fritz working in the plains in the 80s described all of these species in abundance and tied to environmental data, but back then everything was Fragillaria, and now Fragillaria, the genus, is like 20 different genera. So we can use algae base to pull that and update those. We also have open source species descriptions. So this is from diatoms.org, a big collaborative project of, I would say, just about every diatom person, a couple hundred of us out there, collaboratively making what we call an online textbook. So if I go out, collect a sample, I find some species, most of them are going to be on this website. There's almost 1,100 that was updated yesterday, and we're always putting new species pages on there. If I find a species that isn't on the website, I can go in as a collaborator and make one of these nice species pages, names, original names, identification. Maybe you get a nice SEM from somewhere. So we have really consolidated species descriptions, which could be turned into trait data pretty easily. However, we lack consolidated autoecologies and environmental data. It's out there, it's dispersed, it's on somebody's computer, it's in a back room somewhere of a retired professor in paper form sometimes. And what happens is we get this like institutional memory of what this diatom means, sort of based on like who you learned diatoms from, right? So we have this at a conference, I hear like, my sense of that species is this, and it's usually based on like, I can read 6 papers a week and figure out what this diatom means, but it's really not based in primary data. So one of the things I'm hoping to do is consolidate that environmental data to actually have a portal where you can, justify your environmental interpretation by primary data. This is a really good example of an autoecology where somebody has gone through and read a bunch of papers and written it up. Sometimes those have really conflicting interpretations. So this person has both claimed that they find it often in western Montana, but also that this species is mainly tropical. So we can start to see the problems with these sort of like anecdotal, I read a few papers to try to figure out. And then more often than not, really, this is what they look like. Somebody has gone through, clicked a few of the buttons and is like, yeah, it's kind of this big. Maybe it moves around a little. It attaches to some benthic type thing, but it's mostly lacking. We also lack consolidated phenotypic trait data. So this is like a really good version of trait data going on from a pretty pronounced paper studying evolution from one Stephanodiscus species to another, Nyakeri to Yellowstonensis. But again, we run into this like limited access. I have to email Ed Terrio, like, hey, can I use your data? Can you have it? And he's happy to collaborate, of course, but I still have to like go do that. So building a portal to access that would help a lot. We also have a long road ahead, right? So we have this for 16,000, maybe 1,000 species. But if we start right now, we're only in like, maybe 10% of species described. So best estimates are that there's probably 100 to 200,000, and those are the low conservative estimates. I've heard people say like, well, maybe it's more like 1 million or so. And that's just like extant species, so not counting all of the paleontology, which there's a lot. So we have a really good opportunity to start now by consolidating and synthesizing this data. So a vision of what we could know is we have species descriptions, abundance data, we need environmental consolidated trait data, so I want to make like this diatom Rosetta Stone. So we have those 3 data sources, and they just don't talk to each other right now. So basically what I want to do and get ideas from everyone is to have a way for these types of data to talk to each other. So that we could know things about diatoms like the way, Cornell knows about birds, or the way plant botanists know about trees and trait functions. If I was savvier, I would have said and added a slide about how Will and Mason know plant traits. So a little bit proof of concept. This is kind of like what I cranked out with a undergraduate researcher in the last semester, is tying this environmental data, harmonizing our taxonomies, and then having a undergraduate researcher in the lab describe the traits and quantify about 100 species. So that was really great. So we have kind of these 3 things talking to each other now, and that's based on this morphological description book of how to describe the shapes of diatoms. So we have these 3 data sources kind of talking together, but it's really unsustainable. It's CSV files, it's R code, it's really raw and not accessible. Some preliminary insights. The species cluster well in trait space. So each dot here is a species, each axis on the analysis is a trait, and then reduced down to the 2 PCOAs. They cluster well by morphotype, which means we kind of maybe did it right. That's to be expected. And then what's really cool, first kind of glance at this, is that they also cluster well by habitat, which was not any one trait or any one function. So we're suggesting here that now we can, from data, show that like form follows function, or function follows form, one of the 2. So kind of the vision is to design and make this open access platform, run it through the diatoms.org website, and that's kind of what I'm here, to learn from you all, hopefully, about, and write a grant in the next couple of months to carve the Rosetta Stone for diatoms, or so we can have and start to ask these really basic questions like, how does trait shape change biogeochemical cycles, and then make really consolidated paleontology tools. So we can say like the climate was like this, and now, 100 years ago, via trait morphology change, we can reconstruct past climates and environments much better, especially when we run into extinct species. So right now we're just kind of like, it's kind of close to these. But hopefully we can show, well, this trait means that trait means this, and interpret really solid paleontologies. And then we can start to ask questions like, how are traits, what drives biodiversity or biogeography of traits and species there. So thank you, and I'd love to chat if you do any of these things and more. If you saw something in the talk and you're like, oh, I have a tool for that, or we've done this, Thank you.
Thanks.
Or if you just have general questions about diatoms because they're cool.
Hi, thanks. Diatoms are cool. So I was wondering, you talked about developing this online portal and pulling in information. How are you deciding what information is like good enough to be on a portal? Like if a diatom expert has observational data, or does it have to be quantifiable or peer reviewed? What kind of threshold are you calling?
That's, yeah, the hardest question in diatom taxonomy too, right? And then we're like, well, whose identifications do we trust too? Because oftentimes older literature won't have like a picture of the diatom. It will just say we found XYZ species in these abundance, and you're like, prove it? Yeah, so that there's definitely that hurdle. Um, yeah.
Really quick, how long did it take you with your undergrad to develop the proof of concept for the 100 species?
That's been over the last we're on quarter systems, so since January.
Oh, okay, that's
Yeah, luckily that book, The Morphological Guide to Taxonomy, so it's really hard to teach diatom species identification, but it's really easy to show an undergrad, like, this is what a spine looks like, this is what this hole looks like, this is what that shape is, and then they can go through that book. And then she has annotated it down to like, quantifying traits into number data and, using those as a guide. So I think, you know, it's a test run, but in the future, I could hand someone who almost knows nothing about diatoms that annotated book, and they can plug in data. And that's kind of the goal, is to make that really easy to do. So it's actually easier to put the data in the database and get a species description out. Because every summer there's a class that actually it's going on right now in Iowa to teach new diatomous taxonomy. So hopefully in the future they'll use this guide to update the website. The final project for that class is to put a species page on the websites. Hopefully that's pretty quick. Thanks.
Um, the next talk will be enabling biodiversity research in Guatemala with a customized Symbiota portal, and it will be presented by Samanta Orellana. Thanks.
Thank you very much. Good morning, everyone, and thank you for staying to in this session. I am going to present a subject that is really exciting for me. I'm originally from Guatemala, so today I'm going to present about a project that we have been working on since 2020. So I want to present you our own customized Symbiota portal where we are digitizing the collections in Guatemala. We have been working, as I said, with this since 2020, and actually the pandemic helped a lot to realize that we needed to start digitizing the local collections. So with the help of the team at Arizona State University, we started this portal, and it's currently being used as a collection management system for the local collections. It's also a GBIF installation, and we also use it as an aggregator, and I will talk a little bit about this. So one of the things that has made this portal so useful for the Guatemalan community is that it's completely translated to Spanish. So this makes possible for the community to use a lot of interactive tools that we have available to start or complete their biodiversity research. So if you want to visit the portal, there is a QRL code. The URL is in Spanish. It's like biodiversity in Spanish, biodiversidad.gt. So we are a team of Guatemalan biologists. I work with my colleague Sabdi Lopez. He was originally in Guatemala when we started this project, but now he's finishing his master's degree in the University of Southeastern Norway. So along him, we have been managing this portal with a large group of local curators. So all the technical support is possible by the Symbiota team that is part of IDIC Bio. So it's been really helpful for us as biologists from Guatemala that know everyone there to work with the local collections, but it has been very useful for us to have the backup of the Symbiota team for all the technical support that we don't manage. So this project has enabled the collection digitization of more than 34 virtual collections, because many collections are part of a single institution, for example, but the herbaria has, for example, not algae collection, unfortunately, but plant collection, moss collection. So they have several virtual profiles for their own collection. So in total, we have 3 universities working with us. We have 34 virtual collections, 11 observation projects, because some researchers also have images that they want to share with the public and not necessarily want to share it on iNaturalist, for example. They want their own projects in our virtual portal. So we have digitized, I say we, but mostly people in Guatemala have digitized more than 49,000 specimen records so far. And our workflow is really simple. We use the Symbiota portal to digitize and manage, live manage. That means directly manage the collections. They digitize the information from the labels. They take images, they upload them, and the records are instantly available in our portal. So all these records are also available in GBIF very easily with just a click. It's already available in that aggregator too. So this has been really helpful to encourage curators and researchers to share their data. And this is very easy to do because this portal is also a GBIF or an installation in GBIF, and that allows us to connect the virtual profiles directly to GBIF. So we have 3 publishers associated with the portal, 24 collections already in GBIF from out of 34. So not everyone, for various reasons, wants to be on GBIF for now. So we have 42,000 specimens in that global aggregator, and this is very important for researchers and curators because they already have more than 300 citations in scientific literature, for example. And more than 50 researchers are very happy to be in everyone's favorite platform, Bionomia. So this really easy access to digitize and share data has encouraged a lot of researchers, as I was saying, to share their data. And for example, there is this project of digitizing butterflies that have been published in prior scientific papers. So it is really nice to see now the specimens available online and not just the checklist with text in the scientific papers. So this is really, really helpful. And also with new publications, now researchers can upload directly, can digitize, for example, type specimens, can upload them directly into the portal and can share them immediately in GBIF. So that has made things really easy to share type material. And some collections have made a priority to digitize types. So we now have more than 1,000 type specimens from the Guatemalan collections. So unfortunately to me, because I really would love that everything is shared already on GBIF, but not everything leaves the portal. For example, this observation of a very cute feline, this was uploaded to the portal, And then it was published as a new observation. I think it was a new altitudinal record for this species. So this observation is only available in our portal and is not anywhere else for now, but it's published and the information is accessible for anyone that wants to look at it at the portal. So this community is really active. We started from zero almost. Maybe a few collections had a few records already on Excel spreadsheets, of course, and then we were able to incorporate them into the portal, and we have been working with several collections since then. So now we have almost, I think we already passed the 50,000 mark, but this was from 2 weeks ago. So we're probably over 50,000 now. And 85% is already mobilized to GBIF. I am really hoping that the other 15% will be mobilized soon, but that will require funding, for example, or motivation for the curators to do it. Thank you. So one of the important things of our portal is that 92% of the online specimen records from Guatemalan collections are managed directly in our portal. So that makes this platform really useful for a lot of institutions. The only additional collection that is not live managed in our portal is only available in GBIF and is static. So unfortunately, we can't clean the data or do anything for now. So we are not really, our goal is not being an aggregator like for all the Guatemalan data, but we are getting more data to improve our portal in the sense that we need more names for our taxonomic thesaurus, for example, and we need vouchers for the checklist that we can generate in the portal. So we also download more data by request from the researchers. So for example, if we take a look at the data available in GBIF, we can see that all the tiny dots over there should be in Guatemala, but they are not. So that makes us wonder that could we help with the data cleaning, for example, if we import the data from different collections from other countries, maybe we can help georeferencing or correcting the data. That will be amazing. So most of the additional specimens that we have, we use them to incorporate them into this interactive checklist that we can generate. And for example, I have a very cute specimen from Kansas University that I already imaged and incorporated into the portal. So those specimens are coming back to Kansas probably next year, and they will be imaged, and I already have the data to share with the curator so he can update the database here. So last year, we got our first publication about the portal. We have a book chapter published with all the collaborators we have in Guatemala, large group of curators. So it was very important for us to keep working with them and to get this publication so they can also be recognized. So we were very happy because this was a book about biodiversity of Guatemala, and we got citations in 5 more chapters. So that made us think that our portal is being useful for more researchers than our own tiny collections community. So if you want to learn more about our biodiversity portal, you can join us in TED Week in September. We will give a couple of talks over there, and we have documentation in Spanish mainly, and tutorials and everything that you need to know on how to start digitizing your data in a Symbiota portal. So thank you very much for your attention, and I really want to thank IDIC Bio and the Symbiota team for all their help and for allowing us to be here today. Thank you.
We have time for questions.
Hi, thanks for the talk. I have 2 questions. So the first is, I know some collections were hesitant to share their data on GBIF. I was wondering if you ran into collections that were hesitant to share their data to Symbiota and how you overcame kind of convincing them to do it. And then the second part is, so you have about 49,000, if I remember correctly, on the portal. I was wondering if you knew kind of how many collections existed in Guatemala.
Absolutely, yeah. That was one big challenge when we started, right? There were not too many collections that were already digitizing their specimens when we started. So it was kind of easy to get them on board just by showing them that they could digitize their data. They were like, oh, okay, the specimens, they were excited to start and just do it. But the collections that maybe some curators are more reserved and want to keep their data like in local databases, we also have those cases. So we basically, I have to say that most of the curators that have been working with us are my friends, basically. I know them from forever. So it's been really easy to work with them. So that's one advantage we have. But there are several collections, there are several institutions that still like are holding up to see what's happening because there there are other options that they are waiting, like government options that we were actually waiting for 15 years for solutions. So the group that is already working got a little tired of waiting, and some of them are still waiting. So that, yeah. But I'm sorry, what was the second question? Did I address it? How many? Okay, yeah. We only have two large universities that have biology programs. So only those two universities have official collections. And in total, I think we are missing at least 5 institutions. Yeah, yeah. So it's mainly the funding, and they are willing to do it, but they don't have the time, they don't have the funding, and I can't give them. So yeah. Yep, you're welcome.
Any more questions? Okay, thank you.
Thank you.
Using someone's computer is always tricky. Okay, the next talk is Georeferenced Herbarium Specimen Data Improve NatureServe Global Conservation Assessment, and it will be given by Diana Steropoulos. Okay, thanks.
Hi, I'm with NatureServe, and I'm excited to share some of information about our global conservation status assessments. And can I just get a quick hand of who's familiar with NatureServe and global status assessments? Okay, I'm going to go through it a little quickly. It'll be a bit of a review and introduction to some of you, but then also just how natural history collection specimen data improves our workflow, and some georeferenced specimen data summary that's coming to you soon. So conservation status assessments are evaluations of potential risk of species extinction. And we use 3 different elements to calculate this. We use rarity, threats, and trends. And we do this at different scales. There's a global scale, a national scale, and a subnational scale. And subnational can represent native nations, parishes, provinces, states, or territories. NatureServe's conservation status assessment methodology has been evolving since 1978, and it is used by the NatureServe Natural Heritage Network program and the conservation data centers in North America, and our biodiversity data otherwise does extend globally. Conservation status assessments are based on standardized scientific and empirical methodology. We have 2 documents to help us support standardizing this information. And we also use a rank calculator tool to automate the process and facilitate accurate application of the methodology and provide transparency. And all of these documents in the calculator are available for free on natureserve.org website. Conservation status assessment results in a relative rank of summarizing the geographic scale and risk of extinction. For the geographic scale, we have G as global, N for national, and S for subnational, and numbers 1 through 5 that indicate the risk of extinction, with 1 being the highest risk and 5 being secure. And now we'll just go through some quick examples to demonstrate this at the global scale. G1, 2, and 3 are going to be the ones that are at risk of extinction, with G1 being the highest risk level. It's critically imperiled and very high extinction risk due to a restricted range, extreme rarity, often less than 5 occurrences, steep declines, or severe threats. G2 is imperiled, and this is a high extinction risk of a very restricted range, very few populations, or steep declines, or threats, high levels of threats. And the last of our at-risk ranks would be vulnerable, G3, with a moderate extinction risk. And this can have a restricted range, relatively few populations, or recent and widespread declines. Our next two ranks are not at risk yet. G4 is apparently secure with fairly low extinction risk with an extensive range, many populations, but there might be some current concerns about localized declines or threats. And G5 is secure with a very low extinction risk with an extensive population or very extensive range, large population, and little concern for threats or declines. Now that we have some background about the conservation status assessments, we can look into why these really matter. We can use this to identify the most at-risk taxa and ecosystems empirically so that we can make good decisions for our conservation actions, whether that's land management or legal protections for species of conservation concern. And ideally, we want to reevaluate these every 5 to 10 years to see if our conservation actions are making things less at risk or if there are new things that are at risk that we should be providing protection. Our global conservation status assessments for a species are available on NatureServe Explorer. There are over 100,000 species and ecosystems that are posted there. And this will have both the global, national, and subnational ranks for each of those elements and the supporting text for the ranks. When we do a conservation status assessment, we focus our research on 3 different elements. We have 6 rarity factors, 2 threats factors, and 2 trends factors. And for our purposes today, we're going to focus on our abundance and distribution information for range extent, area of occupancy, and the number of occurrences. We collect data from the NatureServe network and the heritage programs, the natural history collections, and iNaturalist. And iNaturalist is completely separate with different concerns for validating data, so we won't get into that today. When we have previously, NatureServe has mostly interacted with the NatureServe network and using their data for our status assessments, but all of the digitized herbarium specimens are so valuable to also include, and so we've been trying to integrate that more. And once we have our observation data collated, we upload it to a Shiny app that thankfully calculates our range extent, number of occurrences, and area of occupancy for us. So in the next 2 examples, it's going to be a map like this. The NatureServe network data is going to be indicated by red, natural history collections in blue, and iNaturalist specimens in gray. Our QR codes are for the NatureServe Explorer links, where you can type in the species there if you're interested in looking at information. Salvia chapmanii was previously included in the very widespread Salvia urticifolia, but it was a resurrected taxon by Keener and Diamond in 2018, and it was included as a species of conservation concern for Florida. So we have Natural Heritage Network data from Florida, 8 occurrences in the southeastern portion of the range. And the rest of our data is through iNaturalist and mostly through herbarium specimens. And we can see if we include all data, it has a conservation status rank of G2 or imperiled. And we really lose a lot of our range extent and our area of occupancy and number of occurrences if we exclude natural heritage or herbarium specimens. So we have an incomplete picture of our distribution and abundance without herbarium specimens, and it makes it look like it's a higher at risk, extinction risk as a G1, G2 without herbarium specimens. Our second example is Monarda austroappalachiana, which was newly described by Aaron Floden in 2015 as distinct from Monarda clinopodium. And it has a narrow range in the southern Appalachian Mountains. And again, it was listed as a conservation of concern species for Georgia. So we have very limited data, just 3 points in a limited portion of the range that also corresponds with an herbarium specimen. And it's the same story that if we have all of our data, it's a G3, a vulnerable species. But if we're missing specimen data, it calculates as a G2 or an imperiled species. So specimen data has a lot of values for conservation assessment and for the NatureServe network. It provides a lot of historical context, including full range extent, where to look for extant occurrences. As this data becomes digitized, there are new areas that might not have been searched by the program. And unfortunately, it also is a documentation for extirpated occurrences that we use with trend analyses. Another benefit is that this data is external to the network because the network likes to work in a lot of pristine locations, and we have a different collection biases through the natural heritage collections that they might document roadside populations that might not be otherwise in the network. And that can provide us with information about threats and trends and intrinsic vulnerability if it's able to persist in habitats that get mowed and have other impacts. So this specimen data is incredibly valuable. I was really excited to bring my experience as the Arkansas Natural Heritage, or Arkansas Herbarium Digitization Coordinator to NatureServe so that I could help with implementing more digitization and having this data available to us for ranking. So in September, I set up a COGI project for conservation assessment georeferencing so we can use geolocate to get this data for STAS assessments. And most of this has been done by a master naturalist in Arkansas who had georeferenced like 10, 15,000 specimens from Arkansas, highly trained and was really on board for expanding her map searches throughout mostly the Southeast at this point. And I'm excited we look forward to sharing this data with you soon. And it represents 43 taxa, over 1,400 specimens, 1,300 of which we georeferenced from 103 Symbiota portals, or Symbiota collections. So that is coming your way soon, maybe in the next week or two. And this is just the start of it. We look forward to working and continuing working on this digitization and sharing that data with you. And I wanted to thank iDigBio for the opportunity to come and present this and supporting me, and I'm happy to take any questions.
I was just curious, like what is the process for new species getting added into NatureServe to be assessed? Because, I mean, for like insects, for example, you know, there's like thousands and thousands of species that maybe only have 1 or 2 known observations. And I guess they have to at least have some kind of threshold of whether they're known at all. So I was just wondering, like, what's the process for that and how it works?
A lot of our species are added because a program has identified it in their state and noted that it's a species of conservation concern. So if we have a state that says, hey, we're recognizing this and we only have it in this limited area, it's an S1, that triggers us to, oftentimes they'll submit a help desk ticket where they want to have the taxon added, and then we'll work with them and the other states through the range to calculate an initial global rank. But a lot of our projects are funded by specific entities, so it can be hard to capture all of the new taxa if they don't fall within BLM or Forest Service lands. And we are trying to work more with taxonomists and with the broader community to make sure that NatureServe's taxonomy is up to date and that we can provide the best information possible for conservation decisions.
I have two questions. I'm curious if when you are doing these assessments, you are checking for misidentification in herbarium specimens, because a lot of times they're kind of just accepted as is, and especially if there's like a disjunct population or something kind of a little bit funny, if there's a process for bringing that in.
Yes, we go through a pretty elaborate process, especially for the rare species of validating the data. For newly described and resurrected taxa, we often go to the original publication, and thankfully a lot of them include a thorough list of specimens that were examined so that we can use the authorities' identifications for inclusion of specimens or inclusion of iNaturalist observations. And for instances where we have like a taxonomic split and we understand the new concept in a certain portion of the range, we remove those disjunct populations that represent a different taxon from the rank and from the consideration.
Any more questions?
Okay, so I'm like choosing which question I want to ask. I guess what I'm curious about is I understand in NatureServe you have the option to basically kind of change the grade of each occurrence that you see. So if you don't see it for a while, you can change it to like not found or something. Mm-hmm. And I was wondering, in terms of taking georeferenced herbarium data, is there like a date cutoff where you're like, okay, this hasn't been visited in 50 years, so we're not going to include this in our analysis of its range because we actually don't know if it's there or not?
We do. It depends geographically where we are located. So for most of the East Coast, we have a 30-year cutoff. For areas that are more remote, it can be 40 or 50 years. And we do try to work with the network on how they are using that evaluation and how frequently they're able to go out and resurvey populations to make those decisions, because if we know that there are tons of populations that were documented 40 years ago in a really hard to access area that is protected and doesn't have threats, but they have made priorities to search other spots, we won't exclude that information if they are fairly confident that the species are still there.
Okay, thanks. Okay, our next speaker is John Adcock. No, David Kunkel. Sorry. Yes, David. Sorry. David Kunkel, and the presentation is Niche Overlap in American Asclepias, the use of herbarium data in broad-scale ecological niche modeling.
So hello everybody, I'm David Kunkel. I'm a PhD candidate at Oklahoma State University under the advisement of Mark Fishbein. This is going to be a slightly modified talk to what was advertised in my abstract, because turns out when you double the extent of your analysis and double the species, the analysis takes quite a lot longer than you think it does. So this is going to be focusing on a subset of the total species of Asclepias rather than the whole system. So just to kind of jump in and give you an introduction to the system, Asclepias is in the plant family Apocynaceae. It's total distribution is across the African continent as well as the Americas, and these two continental groups form sister clades to one another, with American Asclepias arriving and beginning to diversify about 9 million years ago in the kind of Miocene age. Since then, it's diversified into about 130 recognized species in the Americas, showing high morphological and ecological variation. And just a quick note, when I say the Americas, I mean North, Central, and South America, just for clarification. So this group has been studied quite extensively in the realm of adaptive radiation because there's evidence that it rapidly diversified over the course of its history. However, most of these studies have focused on traits such as plant defenses or leaf surface traits, and not so much on the general environmental ecology of this group. So this is where my dissertation kind of tries to fill in that gap. So what I mean by this, just to go through a very simplified example of adaptive radiation via what's called niche divergence. Let's say we have this ancestral herbaceous species on landmass A. We get some propagules from that ancestor that migrate over to landmass B, and it comes into contact with all of these novel environments that it has not come into contact with before. And so through generations, its descendants adapt to these novel environments and form distinct species that we recognize today as the extant taxa, as well as all of the diversity associated with those. So just to kind of think about Asclepias in this scenario, these are maps of the climate and soil zones of North America specifically. And we can see that in the western part of the country, or continent rather, there is a greater heterogeneity of these environments. So if we think about as these environments were forming 9 million years ago when Asclepias was diversifying, we see that there's a lot more room for that kind of niche divergence were it to have originated in kind of the west-southwest of the continent and then spread across the east. So we might expect that the eastern niche overlap might be lower or greater. Is my mic muted? Okay, I'm good now? Okay, awesome. So, jumping off of that, what I used for this study is the largest clade within American Asclepias, the temperate North American clade, and this forms about 57 species of this whole group. And many of you are probably familiar with species in this clade, as there's some of the most common species, such as Asclepias syriaca or Asclepias tuberosa, or more commonly common milkweed and butterfly weed, as well as some of the rarest milkweeds, such as Asclepias prostrata, which is just a very narrow-ranged endemic to the Texas-Mexico border. So we have this great diversity of range variation as well as ecological and morphological variation in this group. So to give you a sense of where kind of the richness centers are for this group, this is a map of the species richness plotted from occurrence data, with each cell representing an area. I believe it's about 40 kilometers for a cell. And we can see in this map that we get these hot spots in the coastal plain, the transition between the eastern temperate forest and the Great Plains, particularly here in Kansas where we are, as well as some spotting in the mid-Atlantic region and some very isolated areas in the southwestern US. So again, just to drive home, there's great ecological variation in this group. They occur in areas that are very desert-like, to grasslands, to forest edges and forest understories. And just because I love this slide and showing off the plants, there's a huge morphological diversity, not only in floral structures, but also vegetatively as well. So using this system, I wanted to ask 3 general questions to get a sense for what's going on in this group. So the first being what climatic and edaphic niche variables differentiate the species in this temperate North American clade. How much do the species within this clade overlap in their niches? And does the amount of niche overlap between species vary with environmental complexity? So is there a kind of a Western bias in greater niche divergence, so lower niche overlap? Or is it not really important? So some hypotheses that go with those questions. Species in the temperate North American clade differentiate along both climatic and edaphic niche axes, primarily focusing on precipitation and soil texture. And then niche differentiation among species in the western United States and Mexico is greater than that in the eastern United States. So to look into these questions, I downloaded occurrence data from these sources, so GBIF, iDigBio, and SciNet. And this was a combination of herbaria specimens as well as community science data, so mainly iNaturalist. And these were cleaned using standard data cleaning protocols, as well as some additions that I made for duplicate detection and uncertainty filtering and several other things. All in all, it was about a 50 to 60,000 occurrence data point data set. So these points were then paired with environmental data, and for this investigation, I used the WorldClim bioclimatic variables, so the 19 of those as well as elevation, and then soil data from soil grids. So these are things like nitrogen content, soil organic carbon, soil textures, cation exchange capacity, and the list kind of goes on. The resolution of these environmental data was about the 1 kilometer scale. It's a little bit less, but it's 1 kilometer as advertised. So using this data for each of the hypotheses, it's kind of like a two-pronged methodological approach. So for the first hypothesis, focused on just seeing which variables are important for the variation in these species, I used a principal component analysis. And then for the second hypothesis, looking at the niche differentiation between the western US and eastern US, I used ecological niche modeling to first establish sort of a map basis for how these species are predicted to be distributed across the landscape. And this was done by generating 10 replicates of niche models for each species, and then taking a model average of those 10 to get kind of a composite average for a given species. And all of these averages were put into a niche overlap analysis pipeline using the Warren's I metric, which I'll touch on more in a second. So how is niche overlap calculated, just to clue everybody in? So we start with average species model. We pair that with another species average model, and we highlight areas that are shared between those two models for positive probability of occurrence. And this is going to then be a proportion of that area, 0 being no overlap, 1 being the niches are identical based on those niche models. So jumping into some results, this is going to be kind of a downscaled PCA plot. And this is probably not what most of you are used to looking at. Most of the time we see PCAs are these ellipses plots that show clear distinctions between groups or not. But as you can imagine, 50-something ellipses on a PCA plot is not fun to look at. So this plot is going to be using the loadings from the PCA plots to kind of look for what variables are contributing as clusters to the variation in this group. So we're just going to focus in on the first 3 PC axes that are highlighted in red, and we're just going to kind of focus in on those. So based on the PCA, we see on PC1, which accounts for the most variation among these groups, that all of the temperature variables cluster very closely together. So based on the percent contribution from PC1, it was somewhere in the 30% to 40% range. Temperature is mostly contributing to the variation in this group, followed up by precipitation, and then following that with soil variables, as listed there on the side. So from this, we could conclude that climate is most important for variation among these taxa, with soil following them up, but not necessarily being unimportant. And just to kind of inject some of the newer analysis into this for the whole group, we get a very similar pattern when we upscale this to over 100 of the species in this group. So the pattern kind of holds. And then going into some of the niche overlap, the next couple slides are going to be species comparisons, just to give you a sense for what some of the more interesting overlap scenarios look like in this group. On the left-hand side of the screen is going to be the first species that's in that list at the top, and then on the right-hand side of the screen is going to be the second species listed there at the top. And then the niche overlap value is right there at the top as well. So, and there's some species ecology information. So, the Asclepias syriaca and Asclepias speciosa, these are these eastern and western US counterparts to each other. If you, depending on which phylogeny you look at, they're either sister or not. But we see that they have very low niche overlap than what we might expect, or maybe not expect at all, just at a 0.16. And this histogram in the middle is going to be summarizing all of the niche overlap comparisons across all of these groups, which comes out to over roughly 1,500 pairwise comparisons. And we see that this sits kind of to the right of the major bars in that histogram, but sitting relatively where the rest of them are. And then Asclepias lanceolata and Asclepias longifolia, these are two sister species that occur in the coastal plain, and this is actually the highest niche overlap value in my data set at a 0.88. And then another set that closely occur in California and kind of the southwestern US, Ariocarpa and Arosa, and this sits very much in the middle of those two previous examples, which I think is interesting. So thinking about Western average niche overlap versus Eastern average niche overlap, we find that Western average niche overlap numerically is slightly lower than the Eastern average niche overlap, but a t-test suggests that they're not statistically significantly different. Some might say marginally significant, but for the sake of this, we'll just say that they're not significant to each other. And on the histogram, Western overlap is marked with the red arrow, and Eastern overlap is marked with the purple arrow. So they kind of just sit very close to each other. It is important to note that with the niche overlap analysis, niche modeling is prone to kind of broad scale distribution predictions if there's too few occurrence records for it to really make good predictions. So a lot of these Western taxa are more rare and thus have less occurrence data, so they might contribute to kind of an inflated niche overlap value. So in conclusion, we see that climate is most important for the variation in this group, and that the Western and Eastern average niche overlaps are not necessarily different from each other. So with that, I would like to thank the members of the Fishbein Lab, Dr. Cody Howard, who helped me with some of the analysis, the OSU High Power Computing Center, iDigBio for providing me with the travel award to come to this conference, as well as my department and institution. And I will take any questions at this time.
We are not going to have time for questions, though. Now we have John Adcock and presenting Make RBGQ Collection Data Globally Accessible. It's all yours.
Okay, well, thank you. Thank you for attending. My talk is very much about program management and implementing a big, large-scale project, as opposed to being more around biodiversity. But obviously, our outcome will be to get the data out there. So I have the absolute privilege to work at Kew and to manage a really large-scale project, one of the largest that Kew has invested into science to date, and equally have the opportunity to work with some really good people at Kew. So anyone who's been there will know that that's the case. I'm guessing it's that. Okay, So we have about 8 million plant and fungi specimens going back to over 250 years at Kew. And of that, about 1 million had been digitized. It took about 15 years to get there. And so there's an onus to actually move on and get the other balance of that done. So we started this project in April 22. It's about £30 million to do this task, and we expect to conclude by March, April 2026, at which point we should be able to have all our data exposed and accessible globally. We have 3 work streams in our program. First of all is to get these non-digitized records digitized. We're now midway through. 3.8 have now been transcribed. About 1.5, just over 1 million are now available globally. The others are in the pipeline getting QA'd. In addition to this project, we've got to build a new integrated collection management system, and we started this in 2021. We've just nearly got all our core data, core collections on there, and living collection and slide collection to be added later this year. And then finally, we are developing a new data portal. We've got the alpha version released, but we've got further work and development to be done on that. So it's accessible, but it's not exposing all our data. So all available data at the moment, both fungi and herbarium data, is using GBIF and being updated each week with what's been digitized. Okay, so why are we doing this? We need to make our data available. We want to be able to speed up biodiversity research and reduce climate change. We also need a catalogue. We need to have a clear catalogue of what we've got at Kew. We have lots of outdated records, but we've got no clear sort of understanding in each box what's available. So that's a critical reason why. And then lastly, we need to provide a surrogate record of our specimens to mitigate against any fire, flood, or pest damage, but also get this data out there. So in terms of setting up this project, what were the main challenges? I mean, I could talk at length about this, but as a nutshell, we've got a 4-year sort of time to get this done. So recruitment and training a team of fresh new staff to come in, to take images, to transcribe or QA the transcription work was a massive undertaking. So all I would say is start early and identify your subject matter experts very early and get them involved in the project as early as possible. And that will allow work processes to bed in quickly. Procuring the digitization supplier, we carried out a pilot working with Naturalis, and that allowed us to understand the workflows, delivery outcomes, and the unit costs that gave a benchmark to develop the project plan, the costings. We spoke to a lot of institutions before we did this, and this goes back a number of years before 2022 from starting it. We went out and did a market engagement of identifying potential suppliers in the market and understanding their capability and experience. And then we invested a lot of time in producing a end-to-end specification of all the deliverables required for the mass digitization supplier. And then finally got to tender stage, where we scrutinized the delivery model, timelines, and what the impact would be that on Kew in bringing in a supplier. And we successfully procured Max Communications Limited, who are actually in this conference this week, or 2 of their directors are. In terms of integrated collection management system, why now? We had about 20 different databases. Some were really old, some were 20 years old plus. They were at high risk of losing our data there, and inaccessible for each. So if you look at herbarium data, you couldn't cross-reference it with seed data. You couldn't cross-reference with the DNA and the tissue. There's a lot of copying and pasting, a lot of downloading data, and lots of faffing around. So we needed to move to get a compliance system, which would operate to relevant legislation. We needed a modern and secure system to mitigate those issues. And we need greater accessibility to view the collection data in one system and link internally and track between collections. So those are the key drivers, and we're now mainstream seeing the benefits of that, and the accessibility of data across the organization is a lot faster and easier. Again, the challenges and lessons learned from this exercise was just getting a dedicated business analyst so you can get and really understand the current, before business requirements and the future business requirements. You can really assess those, understand those. Stakeholder engagement was critical, so we had our senior management team really work across the collection staff so they'd free up time to support the whole process of getting these business requirements and the whole tender process, and then moving into testing the system as part of their work streams going forward. ICMS supplier, again, the business requirement element was just critical in writing and delivering a clear tender specification, so any potential suppliers understood our ask and understand and what we wanted. For us, we needed needed a really effective relationship with the supplier, a collaborative approach. We didn't want a black box. We needed something which is going to be bespoke for Q's collection needs. And that was really critical. So we spent a fair bit of time, part of the tender process, understanding the ability of the system, but equally the relationship we'd get with that supplier. And then we did an end user usability test as part of the process of choosing the supplier. So that was quite a task in terms of the tender process, but it allowed core collection staff to be part of the whole journey and to feed back, so they could get some ownership of what the collection system we were choosing. And we ended up with Earthcape OY, and we've been working effectively with them for the last 2 or so years, and coming to the end of that program of implementation by the end of March 2025. And then finally, the new data portal was acquired at Q. We had numerous portals exposing our data, but they were out of date, uncompliant again. So we needed something which would be UK government standard accessibility, fit that, one system providing global access, and reduce the development time in having to service multiple systems. Now, as I said, our data portal is in alpha version. We haven't completed the developments, but we're underway, and this year we expect it to be operational fully. Key lesson there, just ensure you've got a good development team to accommodate the collections and support them. Okay. All right. So KPIs, benefits, images. All our images are set against the FADGI standards using golden thread to ensure that they're coming through okay. Our supplier has 41 capture stations based at Q, and we have additional 15 or 16 stations because we do our fungarium and palms and orchid collection in-house. So the bulk of the work is done by Max, our supplier, but we're also capturing images daily also. We're bringing in around about 90 to 100,000 images per week. So that's folder data, but also specimen card data and duplicates where required. In terms of transcription, our supplier is delivering about 65,000 transcription records per week, and we at this stage are able to mirror that in terms of the QA work, but we'll be exceeding that to about 70,000 QA acceptance levels per week going forward to ensure we can get everything in by about December 2025, giving you a little bit of wriggle room thereafter to March 2026 when we want to close the program. Transcription is based on a lot of interaction with responsive feedback with the transcription team who are based in India. We support and train the transcription team, and we track each individual transcriber when we're doing the QA so we can understand the quality measures there. Equally, with the image QA, we're very much, we review all the images done of the 41 capture stations in the morning and then again at lunchtime, so we can track if there's any issues. So if anything's going slightly AWOL, if there's any out of focus issues, we can stop that as quickly as possible, work with our supplier, and then review those images which might have come through which need fixing or reprocessing. And those meetings take place on a weekly basis, and we have a lot of interactions during the week with our supplier, so collaboration and the dialogue and communication is really high. we document those so that we're sort of sharing our learning as we go forward. The key performance indicators, we're set by a 4-year period. We have to get this done. We have to get this through. So it's really, we've driven our resources against our timelines. I've given you some numbers. We know we've got to get about 90,000 to 100,000 images done a week to get everything completed. And then that allows the transcription work to follow. So we're looking again about 60,000 or 200,000 transcriptions to be completed each month. And thereafter, we can get in and then QA that and then receive that data in. In terms of benefits of mapping, we set the sort of, it's part of the business plan. So obviously better collection management, efficiency and value for money. We had to do a value for money exercise to ensure was working with an external supplier the right way. Should we review the capability to do it in-house? How many additional staff would we need? What would that mean in terms of time and cost? The other option, which we put in the pilot, was to look at volunteers. How could we use volunteers to actually do this work? Where would we be? And we concluded that volunteers would have to do it a minimum of 7 years, and there'll still be high risk. But equally, to get a number of volunteers in to both do the imaging and transcription work would require so much more space, and it became impossible to do it that way. So comparable cost over 7 years as opposed to 4, but space was a problem. In-house, we felt that we were still not quite comparable cost, and we would have the burden of recruiting and looking after staff. And by bringing in a supplier, offloading those risks was a better and more beneficial way for Q. And so we did the pilot, and we concluded that we were on track to continue on. Clearly, some of the outcomes is attracting government and non-government funding, and then finally, increasing access and broadening the cohort of science research using our data. We're in the early days of that, and as we get and expose more data onto GBIF as initially in our data portal, we'll be tracking the analytics to see how much citations are taking place and where that has increased. So to date, we have 1 million new records to add, or which have been added in the last 6 to 8 weeks, which didn't exist ever globally from Q. So the only way you would have seen those specimens was to actually visit Q. So we're making some headways, early days, but it's coming together. Okay. We have a dedicated project comms person in our team, and we've learned by all the external comms we're firing out, we're actually increasing the awareness of Q science, which is also a byproduct and a great outcome. Okay. So if you have any questions or would like to get involved, by all means, look at our QR code there, and you'll link it to our webpage. But also would like to acknowledge, and some of you may know him and Sarah, Dr. Alan Payton, and they've both been champions of this project way before I started at Q, and architects of the project, and Dr. Sarah Phillips, who's our senior digital collection manager. Thank you for your time.
We have time for one question.
I noticed that most of the data and images for the Q collection that are online are freely licensed, which is awesome. And I was just wondering if you had any insights into how that decision happened and if there was any difficulty like convincing Q to do that.
No, Q have always operated, they want to expose their data for free. That was probably one of the hardest things about getting government funding and others, because they want to see the byproduct and cost. But no, our thing is to be free. Good.
Thank you.
Thank you.
The next talk is the impact of digital data on systematic, phylogenetic, and genomic research. Why designing UCEs probes is necessary for weevil museomics, and it will be presented by Kevin Cortes Hernandez.
Hi, everyone. Yeah, I think I want to start saying that museomics, it's kind of like a, I don't know, fancy word. But I like it because it's kind of like two words united, which is museum and genomics. and I'm also kind of like I like what I do because it feels like Jurassic Park, but real life, because it's kind of like extracting DNA from things that have been dead for some time, sometimes decades, sometimes hundreds of years. And with the technology advancing, we are able to study these molecules that we don't see on the dead specimens. So I want to start like saying that one of the issues of our current era as human beings is that insects, but not only insects, but this talk is about insects, are becoming more challenging to collect or may not be collected again. And there is several reasons. This is like a there's so many reasons for this, right? And the patterns in the last 50 years is that insects are like decreasing a lot. We have seen that this is happening in mammals. I think that 30% of the mammal abundance is being decreased in the past 40 years. Amphibians, I think the diversity is threatened, like half of the amphibian species. But for insects, it's a little bit more complex because how diverse they are, and because we don't know what's going on in the tropical regions of the world. But we have some insights based on some well-studied populations in IUCN, where we have like some population trends. And we have seen that, for example, for these 5 orders, the trend is that the populations are decreasing for 203 species studied for these 5 orders, and also the decrease in other orders, it's just like massive, like 30%, from what we know from well surveyed areas like the UK. That's the B. And then a global index, an invertebrate abundance, says that 45% of the abundance of like invertebrates is decreasing. And this might be like underestimated because lepidopterans are decreasing not that much, but the others are decreasing like more than 30%. So it might be an underestimation. It might be even worse than that. And it's also difficult because we are impacting the world in ways that it's not great for nature, right? Climate change affects insects because they kind of regulate their temperature, so they are depending on environmental factors, and we're just like warming things up. So we're just complicating things with human activities, but also it's getting more complicated because accessing some places, it's becoming more difficult. You can compromise your security to go certain places. For example, I'm from Mexico, and there's some places that you cannot go anymore because it's so insecure. So you cannot collect specimens in those areas. Politics too, it's also another reason. You might not be able to get permits to collect in certain areas, etc. And funding, right? And that's why natural history collections are kind of like a key point for studying insect systematics, right? Because we have knowledge that we have accumulated in the 250 years of our era. And we have all kinds of specimens, dry specimens, wet collections, and we even have fossils that go back to the Dicarian period for animals at least. So we have that available to answer so many different kinds of questions. And we are not only able to study, for example, the morphology, as it was the case in like 50 years ago, but we can also have a lot of information that is being created as extended specimens, right? We can have, for example, I'm working with Symbiota portals. The one that we use for insects is Ecdysis, so I can just do a quick search That's one of the species that I study, Pandeletheus buchanani. And then I can see all the occurrences in different museums where that species is being digitized. And then I can enter additional resources. I can see occurrence records and locality information, and any additional resources associated with that specific record. In some cases can be maps. For example, for this species, we can map all the occurrences in the different collections. That's from the southwest of the US, northwest of Mexico. And for example, if you click in one of the circles, you can see what's the information associated with any specific record, which in this case is habitus images. So for museum work, I think it's important to highlight that when you're working with widely distributed insects, things are like way more complicated, because you cannot go and collect dispersed samples from everywhere, right? So you have to rely on museums. So I'm work with weevils, snout beetles. They have a short rostrum. The tribe that I work with is called Tanimicini, and it has 100 genera worldwide distributed, and almost 1,600 species. So that's quite a lot. And the way to recognize this weevil tribe is because they have this tuft of hairs over here in the thorax. They are supposed to be I think for cleaning their eyes. But it has been studied that these hairs, it's more of an ecological threat. It's a lot of like convergence because of their habits. These weevils live in they feed on the tree branches or leaves, but when they are not doing that, they spend a lot of time underground. So that's why they clean their eyes. But this is a convergence. It happens in a lot of weevil lineages. So that's really troublemaker character to classify things. So there's a lot of things that have been classified in this tribe that are not actually evolutionary related. And we have some studies that says that the neotropical Tanimicini are related to another tribe called Naupactini, based on morphology and some molecular markers. But for the phylogenomic studies, including Tanimicini from the Palearctic regions and the Indo-Malayan regions, it seems that they are everywhere. So the phylogenetics of this group it's messy. And there's other tribes that look really similar to Tanimicini, but the relationships haven't been tested. So again, this is a worldwide tribe. It's everywhere. The neotropical ones, I think we can see a pattern in them. And even so, we can see that this tribe is actually really old. There is some resemblance to some of the faunas from the Afrotropical region, like for example this. I think it would be like kind of like crazy to think that these like similarities are just due to convergence. So I think that there's some evolutionary path share between Afrotropical stuff, but also from Indo-Malayan stuff and Australian fauna. But there's other things that are most likely not to be related. Like for example, we see these. It's clearly that it's a completely different morphology, and it's probably due to these taxonomical mistakes by classifying things based on ecological threats that are not evolutionary linked. And when we focus on the neotropical weevils, we see that Tanimicini also morphologically resembles to other tribes like Naupactini and Anipotactini. And their relationships are not being tested. So I was like, okay, I'm gonna try to do it. My background is in morphology, but I was like interested in getting into genomics. So I was like, why not just try this long shot by extracting DNA from all specimens? So I started digitizing all the specimens available from these collections that provided the specimens to me. That's one thing that I want to talk to you later because there is some difficulties with this. Most of the specimens that I digitized are from the ASU collection where I'm studying right now. I digitized some specimens from Canada collections, but I will talk about that later. And I have 62 genera of the 100 genera of the tribe from all those regions. And well, that's a checklist available in a dices of the DNA extractions that I'm doing for my group. And I'm also extracting DNA from other possible related tribes. But extracting DNA from museum specimens is it's more complicated than fresh specimens because as we live, things start decaying, and when we die, things start decaying even faster. So we have to take that into account. So there's different methods, and target enrichment of certain regions of the DNA. It's one of them. So that's what we are currently using for museomics. And there's certain baits, which are synthetic DNA baits that target biological genomic regions of your data. We have available baits for beetles in general. Beetles, it's diverse, 400,000 species. And there's been discussed that if you use these baits for a highly diverse group for specific lineages of beetles, the target capture is going to be super low. For example, for Adephaga, they use those baits for the beetle baits for the Adephagan beetles, and they found like 21% success capture, which is really low. And also for weevils, it's like 30%, a little bit higher. Okay, so it gets complicated, right? And also when it can be done with fresh specimens, but it gets more tricky with museum ones. So I was in the issue that I don't want to use those baits. I rather design my own. So I've been doing some genome assemblies, and we have different branches of the weevil tree of life, and that's what we're working on right now. And we are also digitizing these specimens and also giving them a new DNA, a new code for this extended information, and that will be available here in the genetic resources on ecDices whenever they are published. And the future directions for my work is like finishing the probe design, which is pretty much computational work, and then test how they do in old samples, because for sure it's gonna be more complicated than if I had fresh ones. And then one of the digitizing issues that I've been finding is that some museums are working with different systems. So how I can do, like, how can I digitize specimens as a researcher? Can I ask the other museums to send me their labels and access to their systems so I can digitize the specimens for them? Or I can just put the data in a data sheet that we can format and then send them to them, which I don't think is ideal. Also, why don't we think of like promote compatibility between different systems? Like here I heard that we're people working with the Specify or Symbiota portals. Why don't we think on like unifying or like at least make these different systems compatible? And thank you. And yeah, I don't know if we have time for questions.
Thanks. Okay, so our last speaker for this session will be Jennifer Kim, with the talk eDNA Explorer, Designing a Digital Tool for Unlocking Biodiversity Secrets.
Okay, hello everyone. We're here today to tell you a story about how we used user-centered design in a field that doesn't see a lot of user-centered work done to create a tool for biodiversity monitoring. And we'll be talking about eDNA. So for those of you that might not be familiar, eDNA or environmental DNA is a way to catalog all the taxa in a local area or look for specific taxa by sequencing all the DNA in a water or soil sample left behind by all the organisms. As a quick introduction, I'm Jennifer Kim. I'm a UX designer and researcher at SliceBread, and I'm co-presenting with Julie Stanford, who's the principal of SliceBread. She's also been able to spin out this project into a company, so she's the CEO and co-founder and also a lecturer at Stanford. And the names on the side here are also the rest of our project team as well. And so to start this story off, we want to introduce you to all the players in this project, starting with ourselves at SliceBread. We're a user research and design agency that uses design thinking to really focus on empathizing and understanding people to solve problems. And so we've worked with a variety of different industries and experiences, with electric car charging to robotic interfaces to even how women shop for sports bras. So it's really eclectic. And we were approached by OceanKind, an organization that funds work towards ocean conservation, to work with Cal eDNA, an eDNA program out of UC Santa Cruz, to really figure out how we can empower more people to use eDNA without needing to be specialized experts. And so we followed our process of design thinking. So we start off with observing what the current situation is through user research, asking people questions and observing their behavior to really get to the root cause of a problem. And then we start unpacking all of that research and pulling insights, figuring out what the key needs are, and start defining what we want to solve for. Then we work with our clients to come up with really creative concepts that start addressing those needs. And then those concepts turn into prototypes, which we then put back in front of our users to have them test it out and observe their behavior, which brings us back full circle. And so we've been following this iterative cycle, refining our prototype continuously until we've gotten to the final product, which we'll talk about in a bit. And so our research started off with 16 interviews with researchers, scientists, and land managers with different expertise and different eDNA experiences. And we sat down with each of them and had a really thorough discussion about their eDNA experience, the current processes that they use, and the struggles that they come across. And what we found is that land managers really want a lot of clarity when they're presented with data. They just want a high-level yes/no on whether a species was detected. They don't want to see a lot of uncertainty, and they really want the data to be focused on their interests. So really their information needs to be unambiguous, clear, and actionable. But the problem was that eDNA is still in its early stages, and scientists and researchers had a lot of questions when they started thinking about using eDNA, like when they should even use it or how to generate trustworthy results. And even those that have used it before had a lot to learn, like how they can identify taxa confidently or if they can estimate relative abundance. And people are really curious about it, but because it's growing so quickly, it's hard for them to stay on top of what's possible. And so because of all of this uncertainty, scientists and researchers had a hard time going back to managers with that really clear data they need. And so because of all of that uncertainty, we decided to sit down and try and catalog all of the challenges throughout the eDNA process. And we found that there are problems in almost every step of the workflow. Just from getting started, there are barriers because people don't know how effective it is or how to make a sampling plan. The fieldwork is the really fun part, but then when it gets to sequencing, people have limited access to labs or don't know what primers to use. Analysis can be difficult because of poor organism assignment from gaps in reference libraries, or if you're not a geneticist, it can be pretty tough. And it's also difficult to look across eDNA data sets and integrate complementary data. And then again, with sharing, managers need that clarity, but because there's such an overwhelming amount of data, it's hard to know what to share with others. And so we really realize that support is needed in these earlier stages of the workflow to generate those clear results. And so we decided to narrow it down to 4 key problems of getting started, sequencing, analysis, and sharing. However, there was a really bright spot in all of the research we found, which is that people really love collaborating. And this really stood out to us because after working with so many different industries and seeing a lot of issues come up because of a lack of communication or collaboration, the fact that everyone in this field loved working with others and actively seek out those relationships made that a really special aspect of this field. And so because of that, we felt that eDNA could really take off exponentially with the right network approach, especially if data is shared effectively. But it's not just the networks that people have with each other. We also realize that there are these 3 interconnected networks with all of the life out there in the world and all of the people in the field, but also all the different biodiversity monitoring methods people use. It's not just eDNA, but also other traditional methods. And so the challenge for us was to figure out how to build something where eDNA can connect into those 3 networks. And so now I'll turn it over to Julie to go over our solution for this problem.
All right, so back to our original diagram, Jennifer took you from observations and interviews to developing insights that she just shared. And then we had a big brainstorm with a bunch of people to really think about how we're going to address that feedback. And came up with a concept that subsequently went through a lot of prototyping and user testing, and I'll talk about that now. So first, the concept. So out of our brainstorm, we came up with a really big idea that had 3 components. First of all, exploration. We thought, hey, since people like to talk about things anyway, let's let people share their projects. So you could see what projects people have done, get inspired to set up your own, and have the whole interface be really easy, obviously. Then we really wanted to smooth out that analysis and sharing by offering access to the best reference libraries possible for organism assignment, offering ways to really easily get to complementary GBIF and geospatial data, and eventually offering some ML-powered analysis tools, and to really help people run a study for their own time, connecting them with labs and giving them great information. And you can see these 3 components really worked across that whole workflow that Jennifer shared with you, from getting started and demystifying to lab partnerships for sequencing to really getting to that high quality analysis and interpretation and sharing of the results. And so then we went to work prototyping. We made a lot of napkin sketches, a lot of grayscale wireframes. I don't have time to walk you through all of that, but we went through that process and I'll show you where we ended up. So this is the homepage for eDNA Explorer, which launched late last year. You can go check it out now on your own. And from the top, it starts off with that first prong of getting started. So you can come in, you can explore projects that people have shared. It introduces you to some kind of keystone ones as a starting point. You can get help running a study, get connected with labs on the platform by reaching out. And when you hop into a project, there's 4 pages of really easy to understand information about the project to dig into. So for example, right at the top, you get project information that everybody needs, who was involved so you can reach out to them, an overview, the location information so you can zoom in and out and see where the samples were collected, and an overview of the methodology so that you can see, oh, this kind of project, it looks like they took this many samples, or here's what happened. Then you get into the meat of it, which is the organism lists. And so on this platform, you can upload sequence data and get really high quality organism lists without needing to interact with a geneticist who you may or may not know, that leverages this really cool Tronco backend and a supercomputer that's running in University of Indiana, and can get you results in a couple days instead of what typically can take over a month. And you can see here there's the scientific and the common name, and there's pictures of each of these organisms from PhyloPic. There's preset confidence levels, there's decontamination filters, and so it really does a lot of work for you to really increase your confidence about these results if you're just getting started, or even if you've been doing this for a long time. And then if you continue on, looking at a project, there's some basic analysis. You can compare what happened over time in a project or across different sites and explore the sites in further detail. And on the point of integrating other data, for every project, we download hundreds, actually, of Earth variables from Google Earth Engine, and some of them we display on the site, calculating alpha and beta diversity for you, and others you can download. And in fact, you can download all of this data to play with on your own. And we have integration with GBIF, and so you can see, okay, here's what I found. Were mourning doves really found in that area, or is that some weird thing in my data? Or maybe they've never been found and I'm the first one. But you can start, again, looking across these data sets and trying to understand how they relate. And when you are ready to upload your own data for analysis, we tried to make a really easy workflow, asking the bare minimum, giving you a really simple spreadsheet to put in your metadata and then upload your sequence files. And then you can also, of course, because we're trying to facilitate sharing, showcase who you are, talk about your skills and expertise. People can look at your profile, and you could look at others. So that's what's launched. Next up, we're working on the fact that now we're getting this really large data set because people are using the site, and we're working with different labs. And so now we can look across this data set and do more analysis that hopefully is going to help everyone across the platform. For example, we can start trying to illuminate organism lists in terms of, hey, which of these organisms are rare or harmful, or are any identified as indicator organisms. We can start looking across the data set to create environmental health metrics and predictive geospatial analysis and map all of that and offer all these tools so that, again, people can analyze their data without the help of a geneticist. So that's us. And if you have more questions about eDNA Explorer, feel free to chat with me. If you're interested in understanding how a user-centered design process can help your product or service work a little bit more effectively, feel free to reach out to Jennifer or to me. And I also wanted to point out, we have museum collections on the site. So if you go there and you look up bycatch in a bottle, for example, that's work that was done analyzing eDNA in different museum samples to look at that whole microbiome of everything that's going on there. So you can check that out. And that's it. So any questions?
Hi there. Thank you. I have like a little bit of a personal love for like human factors stuff. So this was very interesting to me. I was wondering, have you considered or already created any methods and experimentally or observationally seeing what impact this has on efficiency or productivity or even trust, just in general, in sort of this eDNA stuff? You were talking about old timers and kind of not being sure exactly if they knew what to do and could be trained.
Yeah, so in terms of efficiency and productivity, we haven't done any like time studies, but we've done a lot of like user testing in terms of having people come in and watching them observing them using the site, and then iterating on it, which we're continuing to do. And we're getting a lot of feedback that a lot of these processes are easier, but then other feedback that like things are harder, right? Like it's not there yet. It's not ready for land manager use yet. Land managers are going to really need that like more illuminated, like here's the one thing I'm interested in, which is coming up later. So yeah.
So you mentioned that you are comparing data from GBIF, but any plan to develop something to publish data to GBIF?
Yes, 100%. We've been talking to GBIF about having like a little like button literally that'll just send your data to GBIF. So that's in the works. Thanks for asking.
Anyone else?
Hi, thanks for your talk. I was wondering if someone's coming in hoping to do an eDNA project, do you have ways of differentiating, like if the collection was soil, water, air, or if it's single species assay versus metabarcoding, like the different things that are available within eDNA?
To learn about it or to see for a site, for a specific project, what somebody did.
I guess if someone wants to do a project.
Yeah.
Yeah.
So if you want to do a project, you can just basically click like I'd like help running a study, and then that'll, right for now it just generates an email, and then we get in touch. But it's the same process for all those things. Super easy.
And then the projects that are already on there, is there a way to kind of filter them by those things?
Yeah, you can do that in the search. Totally.
Anyone else? No? Thank you.
Yeah, awesome. Thanks.
Hey folks, real quick before you leave, two things. One, reminder that we have the Mexican food truck coming for lunch. They'll be there from 12:30 until 2. Hopefully they will be as efficient as Bryce's barbecue yesterday and get everybody through. The second announcement is the eDNA workshop tomorrow. They said they've got extra space, so if anybody is interested in coming and learning about eDNA or chatting about eDNA, you are more than welcome to join the session. So that's the workshop tomorrow, the eDNA GBIF workshop tomorrow. Okay. Have a good lunch and we'll see you back at 2 o'clock.